Hello, welcome to the August 2016 Get Started with CyVerse. I'm Jason Williams, coming to you from Cold Spring Harbor Laboratory, New York. Welcome. This is the monthly webinar that we have designed to help our new users get oriented to the CyVerse products and platforms. And over the next hour, I'm going to tell you a little bit about the history of the project, as well as give you some information on how to get started, everything from uploading your data to using the cloud. Before I actually go ahead with the presentations, I just want to tell you a few features of the Adobe Connect room that we're using in order to view this webinar. Number one, at the bottom of the screen, you're going to see that there's a place for you to ask questions. And off to my side here, I can see any questions that you may have. So please, at any time, if you have a question, just go ahead and I'll be happy to answer that. That's one of the points of having a live webinar, is that if you have questions, I can answer those directly, maybe even show you the answer. The other thing is that if you move your mouse over the screen anywhere, you'll have the option to adjust the video quality. So if you're on a very good connection, you can leave it at HD. If the connection isn't that good, you can reduce the quality to poor. On the side of the screen there, you should also see that you have the ability to download some files or also click on some links. So there are copies of the presentation, some quick start guides. There's also some links to the various platforms themselves. Finally, and very important, when I start doing the computer demos, is above the screen, you're going to see a button with 4 arrows, and you can actually make the video full screen. And so when I'm giving a computer demo, it'll be easier to see what's going on my screen that way. All right, so first what I want to do is give you a little bit of an introduction to the history of the project. So I've prepared some slides. It'll take us a little bit so you have an overall view, and then we'll do some hands-on demos. Okay, and you can go ahead. Thank you. All right, so the CyVerse project, first I want to tell you a little bit about the vision of the project, and that's transforming science through data-driven discovery. So essentially what the purpose of that is to allow the massive data sets that I think most of us in biology are working with to inform our science, help us to ask bigger and better questions and solve bigger and better challenges. The way that we're doing that, or our mission, is by designing, developing, and deploying really a national cyber infrastructure for life science research, and also training folks how to use it. So altogether, we're more than 30,000 users, over a petabyte of data, hundreds of publications, courses, and discoveries. Now, many of you may be familiar with the original project name, iPlant Collaborative. In fact, we were funded as iPlant back in 2008. The project was successful, and so by 2013, when we were renewed, we became really a cyber infrastructure for life science. And this year, in 2016, we renamed and rebranded to CyVerse, really reflecting that there's nothing plant-specific in the products. It's really open to all life science researchers and investigators. One important thing that I think for everyone using this to know is that we're funded entirely by the National Science Foundation. So it's a $100 million investment that's freely available to your community. We're not a business, we're not a company, we're actually your colleagues and your potential collaborators. Another thing that's of interest to many of our users is that it's really open, and so whether you are in the US or you are based internationally, everyone is able to use CyVerse, and there's no direct cost to them. We also like it when you cite and acknowledge us, because that's how we know we're making an impact. So we also invite you to visit the CyVerse website and learn about some of the publications we have, and hopefully add your own shortly. I've used this term cyber infrastructure a few times, so I should define it. What is cyber infrastructure? Cyber infrastructure, as it turns out, is something that you're already familiar with using, I'm sure. It's having access to data storage, software, high-performance computing, and even people. It's not that any one of these individual things is so unique. It's that we have organized them into systems that really allow you to tackle biological problems that are size of scope that otherwise you just wouldn't be able to do it. And people is an important part of that. So today's presentation is an example where we are actually working to train and working with you as our colleagues, letting you know what's out there, because often the next step down the road to discovery is having a person who knows a little bit about a particular tool or has done a particular analysis before. And so that's one of the reasons we want to reach out to you live today, so that we give you a chance to explore the questions you may have. So these three things sort of summarize it: platform, tools, datasets, storage, compute, training, and support. That is largely what we consider our cyber infrastructure. The other thing that's really important for our users to recognize is that CyVerse is built for data. So whether you're working with plants, animals, you have biomedical data, ecological, climate data, all data sets, because actually whatever type of science you're doing, we're very sure that it will involve multiple data types and integration from multiple data sources. And so the ability to mix and match any type of data set to solve any type of problem is what CyVerse as a cyber infrastructure can give you. This slide gives you a little bit of an introduction to the actual product stack. So there's many different layers to CyVerse, and in fact, different users will want to operate at different layers. For the vast majority of users, they'll be operating at the top here, what we call products. So things like the discovery environment and atmosphere, which we're going to give you an introduction to today, and some other user interfaces, all of which are largely web-based. Beneath that are different services. When you log into CyVerse, you have your data storage, and it's unified across all of these platforms. And when you are going ahead and searching for data, or you are logging in, all of those things are under single unified services. When we talk about actually the cloud or the different computers, if you're a developer, you can actually access those things through the APIs. So this arrow here, ease of use, when you're operating on the web, most bench biologists would probably place themselves here. When you're actually operating at the level of systems, and you may have really large projects, you can also access these things as a developer. A really quick introduction to how CyVerse is built, and so to wrap that part of the story up, I'd want to say that we've known for some time in science that these big data challenges were coming, and we also knew that it wasn't a matter of just a few individual people solving the problem, but really gathering the community together to find out what the requirements are. And so the first 1.5 years of the project were really largely focused on determining from the community what were the needs. Out of those priorities are actually the same things that we see that are important several years later. So genomic and genomic data are still the big drivers of what are the big data analyses within biology. And so when you come to CyVerse, you'll see that you have tools and services that will enable you to do everything from reference-guided assemblies, RNA-seq, variant calling, ChIP-seq, and really a large set of things that having to do with the manipulation of sequence data sets. The other thing that's important about CyVerse is that we're completely a collaborative and virtual organization. So CyVerse is based across these 4 institutions: University of Arizona, Texas Advanced Computing Center, which is part of UT Austin, Cold Spring Harbor Laboratory, and UNC Wilmington. So all of these organizations collaborate and provide different pieces to the CyVerse platform. CyVerse is also established in the UK as an independently funded project, and that's a collaboration between several universities and institutes funded by BBSRC. Okay, so let me just stop here and give you a quick sort of a 10,000-foot view. What you want to think about, if we draw an analogy, is that, well, the one that we are using here is this idea of Lego blocks. We have built 85, maybe even 95% of the pieces that you need, the various tools, the various services like data storage, and then what we help you to do is assemble those pieces into the workflows that are suited for the type of analysis that you're doing. So what I'm going to do in these next set of slides, and then that's going to bring us into actual giving a hands-on demo, is tell you a little bit quickly about the different pieces. Some of them will actually have demos on, and other ones are just for you to know that they exist. Okay. So the data store is at the basis of this. With the data store, you actually have the ability to look at a 100 gigabyte allocation for each individual user. And if you need more data storage than that, you can certainly get it. Terabyte allocations are available. Just give us a little justification. The data are automatically backed up, and it's really easy to upload, download, and share. And I'll do that in my first demo, give you an idea of how that works. The next piece is the discovery environment, and this is a web application or web interface to hundreds of bioinformatics tools. And it's really a platform, and that means that you can run any bioinformatics application that you'd normally run perhaps at the command line. It's integrated with our data services and high-performance compute, and you can even add your own applications. We'll do our second demo on that. Atmosphere is our cloud compute resource, and if you haven't used cloud computing, we'll introduce to you why it's a really tremendous resource for anyone doing computation. The science APIs are wonderful for folks who are developers and want to directly access various parts of the cyber infrastructure at that level, and you can certainly develop programmatic resources using science APIs. DNA Subway is a platform for educators, so if you are interested in bringing bioinformatics into the classroom, DNA Subway has several what I would call classroom-friendly workflows for analyzing data and even developing research-based activities in the classroom. And finally, BISC is the other large piece of the puzzle. This is our image analysis management platform. So you can handle hundreds of different types of image formats. You can analyze and manage thousands, tens of thousands, millions of files using BISC and the powerful routines that have been developed within that. Okay, so that was our pretty brief introduction into what the CyVerse platform gives you. It's really important for our users to know what the project goals are and where we came from a little bit. And what we're going to spend the rest of our time in is on actually giving you some hands-on demos. For all of these demos, I'm just going to sort of walk you through. Some people might follow along, but probably most people find it beneficial just to sort of get an idea of what's there. All of these things are very well documented, and you can follow along at your leisure. Additionally, the entire presentation is being recorded, so you'll be able to go onto YouTube later and rewind and look at anything you missed. Let me also remind you that at the bottom of your screen, if you have any questions, off to the side there I can see those questions, so please at any time just let us know if you have any questions. Okay, so let me go to the demo, and this might be the time where you want to make your video full screen if you haven't done so already. There's some button at the top there with the 4 arrows, and you can make it full screen so you can see my computer a little bit better. Okay, so quickly, the CyVerse data store allows you to store any type of file related to your research. You can move the files seamlessly between the different platforms. You can automate your transfers, and you can even share your files with lab members, with the community, with collaborators, whomever. There are several different methods to access the data store. I'm going to show you the point-and-click methods involving CyberDuck, which is a standalone tool, and the discovery environment. I'll also show you the command line method at the terminal for those of you who'd like to do it that way. In terms of why you would use one or the other, the discovery environment is the web interface. I would recommend that really for sharing the data. There are some limitations to the size that you can move within that platform, but it's really easy to use. For general purpose, just about everything, if you have a 300 gigabyte data set that's sitting somewhere and you just want to drag and drop it, CyberDuck is going to be the first thing I'm going to show you, and that's really general purpose and works for moving large data sets relatively quickly. And iCommands, if you require programmatic access and want to do things at the terminal, you can definitely use that. Okay, so let me show you on my computer how some of this stuff works. I'm going to go ahead and open up the website for CyVerse. And while I'm here, and let me just get that formatted for you a little bit better. While I'm here, let me remind you that when you have questions, you'll want to come to the Learning Center, which is here. And from here, you can start to get access to the tutorials and documentations that you need. We're going to start with the data store. And so everything that I'm talking to you about now is going to be here. One point that I emphasize in terms of performance is that we'd expect, in terms of transfer, about, there's in a sort of a little summary, about 100 gigabytes moving in 30 minutes. So in a good connection, you can really move data in and out of CyVerse really well. These are all covered in detail here so that you can find out about that. So please read through that when you have a chance. Right now, I'm going to go to this section, Manage Your Data Basics. and it's going to take you through some of the methods. So the first one I'm going to talk about is CyberDuck, and if I click there, it will give you the documentation. CyberDuck is a standalone tool, so you're going to have to download that. I'm just going to wait for that to load because I actually already have CyberDuck set up. But really, it's available for Mac and Windows, and once you download it, you just need to follow these 3 to 5 steps depending on where you're at to get that started. And here's what it looks like. I already have it running. Okay, so you start CyberDuck, and the very first time you run it, there's a configuration which allows you to import or to actually log in and add your CyVerse credentials. But what I'm looking at here is actually the contents of my CyVerse data store. So these are all the contents. And if I want to move a file or folder, it's really as simple as dragging and dropping. So let me find something that's not too big. Okay, here's a folder, VCFs. And if I want to move that to my desktop, I simply drag that to my desktop. There will be a dialog box that opens and tells me the status of the transfer, and within a few moments that will start downloading. If I wanted to upload a file, and let me just on my desktop create a test file. Okay, here's a file that doesn't really have anything on my desktop. But you can imagine this could be a folder, it could be a file with whatever data set, and then moving that into the CyVerse data store is just as much as dragging that over, and it will allow me to upload that. And again, down here at the bottom, I'll see the status of that if something was interrupted or moved or whatever, and that's really easy to do. Okay, there's really fortunately and unfortunately, for the sake of the demo, nothing more to show you. Once you downloaded CyberDuck, and again, let me go back to those instructions on the wiki, you'll just find the link to CyberDuck to download it for whatever your platform is, and it's currently available for Windows and for Mac. Once you download it, there's a second download that you need, which is here, which is the configuration of the connection profile. So that's like a bookmark. If I disconnect from CyberDuck, or actually let me click disconnect, you'll see that once it's set up, it goes back to having the CyVerse bookmark. So waiting for that to happen. Take a second. But really, that's really, you just download that second thing, it'll automatically open, and you'll follow the directions here, which will tell you how to enter your username and your password, and just another connection protocol to enable you to move the data a little bit faster. But that's about it. This is taking its time to disconnect. But that's what it will look like once you have it all set up. You'll click that bookmark again, and it will start logging you in. In my experience, it takes about 15, 20 seconds. But that's about it. Moving your data to and from CyVerse is going to be as easy as opening CyberDuck and dragging and dropping. All right, let me show you one more method before I go to another point and click method, and that's at the command line. So let me show you where the instructions are for that. Back on the Manage Data page, there is a login to iCommands. So it'll tell you with a CyVerse account, and it will go through the process of showing you how to install iCommands. It is available for Windows and also for Linux. So you can follow the instructions, and if you're familiar with the command line, it won't take you very much to get started. I'm just going to open up a terminal. It's a little bit easier to see. The very first time you run it, you'll run a command called iinit, which logs you in. I've already set this up, so it's just going to accept that. When you do it for your very first time, follow the directions, and it will tell you to enter things like the port, the zone, and your password, and you only need to do that once. Once you have actually connected, then it's very much like Linux. So for example, an ILS lists the contents of your data store, and these are what we just saw a few moments ago in CyberDuck. If I wanted to download something using the command line from my CyVerse data store to my desktop, it would be as simple as an iGet. And then if I want to get a folder, I'll have to make it recursive. I want to see the progress, verbose, and I want to get .vcs. And I could download it to my desktop and hit enter. And there it is on my desktop, along with the progress of everything that's happening there. And that is as simple as moving the files down, and an iPut would put files back up. But you hopefully get the point just looking at it, that moving data from your local device up to CyVerse is really pretty simple, pretty robust. Okay, now let's show you one more thing, which is actually how to share data. And to do that, I'm going to go back to the CyVerse webpage. And then we'll actually open up a new tool that we're going to talk more about for analyzing, called the Discovery Environment. So from here, I'll go ahead and click on Discovery Environment. And down towards the bottom, there's a little link called Launch the DE. So I'm going to go ahead and click on that. We'll talk a little bit more about the Discovery Environment in just a few moments. But for right now, when you log into the Discovery Environment, this is what it looks like. And I'm just going to click on this first window called Data. And if I look, I have a folder there that is the same as my CyVerse username. And that's my home folder. And again, I see all of the folders and files that I was just viewing in CyberDuck or viewing at the command line a few moments ago. So from here, there are some additional ways to import data. You can upload things, smaller files from your desktop. It's not covered here, but there is actually an application. We'll talk about it. We could mention if there's a question to import data directly from the NCBI SRA. But what I want to focus on with the Discovery Environment is actually sharing data. So from here, if I have a file or folder I want to share, so here's a file. For sharing files, not folders, but files, I do have a quick shortcut. Under Share, I can say Create Public Link, and click on that file and say Create. And it will create a URL that I can share with anybody, and they can download this. So if I click on Copy Link, and then I was to open up a web browser and click that Enter, then this would allow anyone to download those folders or files. Rather, I'm sorry, not folders, but files, individual files, just from that link. Now, it's worth mentioning that this link, as I said, is restricted. You won't be able to share folders in this way. However, you can compress items. And if you have relatively small, so I wouldn't share gigabytes of data this way because often over the web, transfers are slow, and there's a better way to share large data sets. The other thing about this way of sharing is that it's insecure. Anybody who has the URL would be able to download that. And so once you're done sharing, you may want to come back to this and just click on this little delete link. And now the file is no longer accessible through that URL. If they've already downloaded it, that's fine, but you can have controls over your links in that way. Now, if you want to share a larger file or even folders, what we recommend is that you use the sharing feature that allows you to share with collaborators. And what this means is that I can share any size data set with anyone. I may have, 500 gigabytes in this folder. All I need to do is go up to Share and say Share with collaborators. Now, this does require that they do have a CyVerse account, but as you hopefully saw, it's not that difficult to get an account for free. Then I can just type in their name, excuse me, and I should be able to see their name and their institution. I click on that. And then I can decide what level of sharing I'd like to give them. Now, if I go back to the documentation here on managing data, let's see if I have that. Or I can actually, since I'm in the wiki, click on sharing files and folders. So the wiki is one of the ways we have this documentation. If you go through this, there is a little bit about sharing the folders and the permission levels. So there's a little chart that probably will help us. These are like Linux permissions, if you're familiar. So if someone has read permission, they can read files and they can even download files. If someone has write permissions, they have the ability to read, download. They can adjust some things like renaming. And then finally, if they have own permissions, they can do everything, including moving and deleting. So you need to be very careful. Don't give own permissions to somebody unless you're prepared for them to delete, even accidentally, your files. So once you decide what level, you can click Done. They'll get a notification it's been shared with them, but I'm not going to do this right now, so I'm just going to cancel that. In fact, if this was already shared with Cornell in this case, I could open this again and just click that X to revoke those permissions. Okay, and click Done. When something is shared, it appears in the Shared With Me folder. And if you click on the Shared With Me folder, you'll see the usernames of several users or services that share data with everybody, but you'll also see the links to something that was shared with you. Final thing that I want to talk about, and this is certainly not everything I could show you. For example, with files and folders, there are other functions here to rename, to move. Most of it is quite intuitive, so I'm not going to spend a lot of time going into that. But I do want to talk about searching. So I can certainly search for files, and if I search for a genus, maybe I pick up some things that come up with that particular genus name that I've worked on. But you also, with a file or a folder, have the ability to assign metadata. So if I click on a folder, or rather in this case a file, and go to metadata, I might not own this one. Let's see if I do. That might not be giving me what I want. So let me go back to something that I have actual permission to. Maybe one of these. All right, let me go back to a home folder. If you select something, you can go up to metadata and you can edit the metadata. Now, I don't have time to give a really nice full lecture on metadata, but essentially with metadata, it has an attribute of value in unit, so I could have an attribute like time point. The value might be 1 and the unit might be hour. But as I'm doing experiments, and let me explain this for just a moment. When you're doing an experiment, you probably can't simply name your files or your folders according to every detail of the experiment. So it can't be, experiment underscore genus underscore trial one underscore sample one dot txt. Ultimately, that gets too complicated for the sizes and the types of data sets that we're working with. So within CyVerse, you have the full ability to describe metadata for any particular file or collection of data sets. And what that means is you can even, if I'm not going to show it here when we call bulk metadata, is you can have all of your files and folders. You can have a spreadsheet, essentially a CSV or some type of tabular data format that describes your data set, and you can pair those in the discovery environment, which makes it super powerful. So let me go back to my computer. This that I'm showing you here is just a way to look at it very simply. More useful is templates, where I can say, okay, this is going to be a DOI request. And if it's going to be that, there's a template for the types of things that I need to have associated with this data set. And I can fill out the template, in this case manually, but you can also do it in bulk. Once you have metadata, I'm not going to save this, then you can not only search, this way, but you can click this funnel, and now you can search more specifically. So for something that has the value genus, and it's genus Dioscorea, now I can search all of my data sets, and I can have really, really good control, fine control over what data set will come back in any particular search. And I can even save my searches and create custom folders so that every particular file that matches particular attributes and values can be placed in one or another location that I can easily find. I'm on wireless, so this is taking a little bit longer than usual, but hopefully get the idea that you can go back and you can search according to any particular set of values that you might describe in your metadata. So it's quite powerful. Okay, so I don't have much more time to go into that, but hopefully you take another look. And hopefully you've seen that you can also, if I go back, one last thing I'll say is you can create your own folders, you can share those folders with other users, and so now you have a shared space for data within your lab. Okay, so I have to move on. I have a couple more slides, and then I'll talk to you about how to actually analyze some data. I'll go back to my PowerPoint. Okay, and this is going to be within the discovery environment. So the discovery environment, as I mentioned, is a platform. You can run almost any bioinformatics application, and it's integrated with your data and high performance computing, and you can actually add your own applications there. Using the discovery environment, there's really those three main windows. We've already shown data, and you know what that can do: upload, download files, share links, share files and folders. Now we're going to talk about apps where you can actually run any of the hundreds of bioinformatics apps that are there. We won't have time to integrate a new application or build a workflow, but those are described in documentation. And then also under analyses, we'll view the status of a job and see a history. Okay, so I'm going to give you a very simple demo. In this demo, we're just going to create a multiple sequence alignment using muscle. And we're going to take unaligned DNA sequences in FASTA format and just create a multiple alignment. So let me go ahead and show you how that works. Once you get the feeling for how this works, then you'll basically be able to use any application that's there. If I go on data and I just want to take a look at the data, I know that this data is in, actually it's one of my favorite folders. So you can favorite folders, you don't have to keep looking for them. It's in this place called DE Walkthrough. And I have this text file called DE sample plants, and I can click on that file just to preview it. You can open various types of files within the discovery environment. It will automatically format tab delimited or comma separated tables, FASTQ files you can view by page so you don't overload your computer. So here's some unaligned sequences. And if I want to align them, I'm going to go ahead and use muscle. So I'm going to close the data window and go ahead and open applications. Okay, this is apps here, and from apps I can go ahead and search any of the applications that are located within the discovery environment. From here I can type in the name of the app like muscle, and from there I can choose an application. Now there are different versions sometimes. If you have one that you like, you can click the favorite. If you're not sure what an application does, you can click the info button, and that will give you a short description. You can also click on the user manual to see more about that application. If an application is listed in red, that means it's unavailable for some reason. Sometimes that may mean that the system that runs that application is on maintenance. Sometimes it may mean that the application has been deprecated, but often it's possible for you to use it. You'd have to contact support or post a message to our user forum. But I'm going to go ahead and just use an application. So I'm going to click on the name of the application, muscle. And I can close the catalog since I'm not going to come back to it. And every application or analysis has a name, so I'm going to do _demo. I always encourage you to put comments, even for something that's very simple. So this is a demo for the webinar. I have a chance to choose where the outputs of this analysis will go. I'm going to leave it by default, which is this folder called analysis, but I could change the default up here in my preferences. I could say, you know what, I have a folder that's dedicated to my lab, and so I could make that one the default and all my analysis would output to there. Okay, but for right now, that's all the changes I want to make. So I'm going to move down the menu here. It's kind of like an accordion. So the next one is select input data, so I'll click browse. And it's going to move to wherever I was last time, so I just need to move it back, and I believe this is an iPlant training. And then DE walkthrough, DE sample plants, and say OK to that. And as I look, the next menu is sequence type, and I do know that that is DNA. Okay, so I'll go ahead and click Launch Analysis now that I've moved through all the steps. At the top of my screen, I'll get a notification that that analysis has been launched. And now I want to probably see what's the status of that analysis and the history. So I click on Analysis, and that's the last menu there. Okay, in the Analysis panel, what I have is a detailed history of all of the analyses that I've done using the discovery environment. So I can click on any one of these and get details. Right now, this analysis that I just launched a moment ago is listed as submitted, so it's not ready yet. If I click on the analysis, I do have the option, since it's not done, to cancel it. But right now, what I'm going to do is click on View Parameters, and I can actually see details about this analysis. There's a link to the actual file, so if this was 01100002.fas, which has no meaning and I wasn't sure, Clicking on that link would take me to the actual file, and I could take a look at it, so I don't have to guess. Also, all of the parameters that I selected, like it was a DNA sequence type, are already selected there for me. I could save this as a text file. So basically, this is like a really detailed lab history. If I were to actually click on something that's a little bit more complicated, like a top hat alignment, and view parameters, again, now I see all of those files, I see all those parameters, and I don't have to guess about how I arrived at an analysis, so that's really good. If I want to relaunch an analysis, rather than clicking lots of buttons and refilling parameters again, I could go up to Analysis and say Relaunch. And that would allow me to look at that same application. So this is an application we haven't seen before, Top Hat, except this time when I click on Input Data, all of my files that I used in the previous one and all of my settings are exactly what they were the last time I used them, since I said relaunch, okay? So that's quite useful when you have to go back and redo something. You don't have to do all that clicking, copying, pasting. So back to analyses again. I'm going to click refresh to see what the status of my analysis is. I didn't get any notification yet, so I assume that it's still in the process. I'll give it a second. Okay, it's still submitted, so it'll take a few more moments to go ahead and work. What I'm going to do is click on an analysis that's done. So once it's done, you click on the analysis name, and it will take you directly within the data store to the outputs of that analysis, in this case, which is some aligned sequence. And give that a second to load. And these are my aligned sequences. Okay? So really, once you understand the idiom that's there of you have some data sets that you've moved, you positioned, and then once you have those data sets, you can go ahead and use the apps. And currently, I didn't mention it, but there's several hundred, so it looks there's 437 applications that you could take a look through. Once you launch an analysis, you'll get notifications as you see, okay, this analysis is run and completed. In fact, if I open up analysis again and click refresh, the analysis that I started a few moments ago is now done. So I'll refresh that. And once an analysis is done, you can click on an analysis name to go ahead and see the actual output. Okay. So basically, that is a quick summary of how the discovery environment works. You have the ability to take any one of those applications, load your data set into it, and then you have the ability to use those applications to launch an analysis, even create a workflow that we can show it to you, and then you can go back and monitor that. Very detailed notes are kept in the system so that you can always reproduce your analyses. So it's really a useful platform to get started analyzing your data. If you take a look at several of the tutorials for working with genome assembly, RNA-seq, or whatever you're interested in, then you can go right ahead to the discovery environment and start analyzing lots of different types of data. Okay, so I have one last demo to share with you. Let me remind you that at the bottom of your screen, if you have any questions at any time, please go ahead and send me those questions. Haven't gotten any yet, so I assume that you are pretty much okay. The last thing I'm going to show you is on cloud computing. So let me go ahead back to my slides, and I'll show you a little bit about how to use Atmosphere. Okay, so this is the last section of what we can show you today. And Atmosphere is what we call cloud computing, and it gives you access to hundreds of these virtual machine images. But there's a little bit of jargon. If you've never used cloud computing before, you may not know what it is. So let me explain it by means of an analogy. I think we've all been in the situation where we have an old computer, and then we buy a new computer, and so we'll need to move our data from the old computer onto the new system. So we take a hard drive, we copy our documents and our files, data sets over into the backup drive. However, it may be the case that you want to make a different type of backup. And so there is a file, and this is the word that you're going to need to remember. The special term is called an image. You can make this file called an image. And an image is another type of file, except that it is a complete copy of everything that is on the original system, including the operating system. So now on your new computer, if you take documents off the hard drive, you just have those original documents. But if you take an image file, you have everything. So you would not need to reinstall new software. The software is already installed. The operating system is there. Everything that you set, every preference, your desktop image, photos, all of those things are exactly the same. And so that's what we call an image. An image is a file that contains the operating system as well as settings, parameters, and installed programs. Okay, there's one other piece of vocabulary, and that's called an instance. Okay, in the CyVerse cloud, we have hard drives, we have CPUs, we have memory, and for anything, any type of job that you want to run, you're going to need a certain amount of those things. So you can request what you need, and then you also need to grab an image because you need an operating system to run a computer. And once you have all of those, you actually have what we call an instance. And this is a virtual machine. That's another term for it. And it's available at this IP address. You'll be assigned one when you use Atmosphere. So what's the value of this? Well, there's lots of different things. One, you can choose any of the operating systems with the software installed that we have, or you can actually build your own. You can get systems that are pretty decently sized, up to 16 cores and 128 gigs of RAM. You can access things via the shell or VNC, and you can actually share your images with other users. You can even make them public. So there's lots of advantages. You can run software or analyze data sets that right now you're just exclusively doing it on your laptop. In the discovery environment, you can't run anything that has a graphical user interface. So if something already has an interface, you can run that potentially on Atmosphere. You have full administrative or sudo access on your virtual machine in Atmosphere. You also, if you're working in a classroom or a teaching setting, it's wonderful because you can create a uniform setup. But even in your lab, you can make sure that every person using any type of analysis in your lab is working from the same set of software and parameters. And if you develop a software, script, whatever, you can actually use Atmosphere as a way to disseminate that. So I have time for a quick demo, which is just going to be how to launch and connect to Atmosphere. So let me go back to the CyVerse homepage and close some of these windows. Whoops, CyVerse. Okay. From the CyVerse homepage, you can go ahead and click on Atmosphere, and then you'll see the link there to launch. And this is Atmosphere. This is the dashboard. I won't go into too many details now that aren't super important, but it gives you an idea of what resources you've used. It also tells you what things you've launched in the past. Then I'm going to go ahead and say I want to launch a new instance. And I could search many different ways. So for example, let's say I was interested in doing RNA-seq. I could see what software and pipelines people have installed. So there's various different ones that folks have, and reading the description, I could take a look and see maybe some of them are useful to me. Once I choose one, and let's say I choose this one, I can go ahead and click on Launch, and then it'll take me through the process of figuring out what I need. So I may need a certain number of CPUs, depending on what I have access to. Oops, let me go back here. I have that full capability, although you have to remember that you have an allocation. So if you go over your allocation, you'll need to request more resources. Also, every single, especially the larger machines, are not available every single moment. If they're all being used, then you'd have to use a smaller one. And you do get a certain number of resources that you can use up to each month. So after the month is over, it will refresh. And again, if you need more, you can request more using the buttons that you have on the control panel. I've already got one launched, so I'm not going to launch this one, but it would just be a matter of clicking launch to get that started. So I'm going to go ahead and click cancel. I'm going to go ahead to a project where I've already had something running. Okay, so I have some virtual machines running, and when you launch them, it takes somewhere between 10 and 15 minutes before it's available. You get an email usually when it's done. And when the status is green, that means I can connect to it. Okay, so let's see, I think I launched this one just a few minutes ago. No, that's the older one. So let's click on this one I launched a few minutes ago. I have some details about it. The most important detail is that it has this IP address. Now I'm going to use a software called VNC Viewer. this will allow me to connect to my Atmosphere instance. So some Atmosphere instances have a desktop, in which case you can use VNC Viewer, and they all have a terminal, and you can use the shell or terminal installed on your Mac and Linux. Now that Windows is getting Bash, maybe you can use that too, but you can certainly use PuTTY. So let me go ahead and open VNC Viewer. And just following the documentation, all I need to do is paste in that IP address, and then also enter a :1, and then click Connect. It'll say, do I want to connect? I can say yes. I enter my CyVerse username and my CyVerse password, and click OK. And this is my desktop of this remote computer. And I can really do anything on that desktop that I would on a normal computer. I could go ahead and start an RStudio session. I could run a platform for analyzing some data. So really anything that you could use a computer for, you could use this temporary virtual machine for. Okay? If I want to connect to this using the terminal, I can do the same thing. In this case, I would use SSH, my CyVerse username at that IP address. I can say yes, connect to that, enter my CyVerse password, and I'm also connected that way. If I want to make a directory on the desktop, there's my directory on the desktop, okay? But I can connect to this virtual machine and now do anything I wish with it. When I connect for the very first time, it's not connected to my CyVerse data store, so I would have to connect it using, in this case, I could use iCommands, which is installed. I could do that quickly if I wanted to, IINIT. I think I remember all of these things. Okay, and now I'm connected on that remote machine, and I could bring data onto my desktop. Okay, and then on the desktop of that new Atmosphere instance, I brought over something from my data store. Okay, so that's really all documented. When I come back to this interface for Atmosphere, I can see the status of things that I've used. If I need to reboot, I can do that. And then finally, when I'm done using this, I want to either stop it if I'm just done temporarily, or if I'm really done using it and I'm not going to come back to it, I need to delete it. You need to do this because if you're not using something, we want it to be used by folks who need the resources. And eventually, your resources will start to spend down. The key thing is that you must move or save any data off of this virtual machine before you're done. So you can move it back to your CyVerse account. You can create a drive within Atmosphere. So that's described within the documentation. All those things would allow you to preserve that data. Okay, so I am not going to use this. I'll go ahead and delete it. It gives me that last warning, and I'll say OK, and we'll start to delete. Okay, so that is essentially the end of the demonstrations that I wanted to give you. The last thing I wanted to draw your attention to is that we have a learning center that you have the ability to go take a look at that gives you additional information that allows you to get tutorials and even see upcoming workshops and webinars. So I'll take you to that in just a second, although right now I have a question by Brianna who asks, how well does Atmosphere tolerate usage by group of people? If we wanted to use one instance with your undergraduate lab. What our recommendation would be, Brianna, is that actually you have every student have their own instance. You can share, but then it's almost like sharing a laptop. I mean, so if you have some activity where it's okay for maybe 2 people to share a laptop, but all of a sudden if you have 10, if you have 10 people that you are working with to share, it becomes confusing. We actually have a program called Atmosphere for Education, so you can actually book time, tell us how many instances that you need, and we can help you so that during your class time you can actually use it. So you can contact support or cloud at CyVerse.org, and we could give you some more details on that, but we recommend that everyone have their own. Let me just go back to the website just for a moment, And if there are more questions, please let me know. Okay, so back to the website. The last thing I want to show you, and then we'll be finished for today's presentation, is I've mentioned the Learning Center. So please take advantage of the tutorials and resources there. Let us know what else you want to see. But also know from the homepage, I'll just go to the bottom, there's a quick link. There is also the Ask link under Connect here. And from Ask, it's actually our user forum. And so if you have questions, you'll probably be able to find someone who's answered them already. There's almost 1,400 questions. But if your question is not there, the community members and also your various colleagues and our staff will be able to look through and help you with some of those questions. So with that said, we thank you for joining us for the last hour here. We hope that this has been a useful introduction. We know that many of you hear about CyVerse for the first time, sort of word of mouth. When this webinar completes, we'll push you to a survey. I believe it's updated to include today's presentation, so please leave us some feedback. You'll also get a notice once the video is posted to YouTube, and you can take a look at that there. So I don't see any other questions, so I'm going to thank you for your time. Thank you for signing up for your account, and we hope that you really get a lot out of it. And we thank you for signing up for CyVerse. So have a wonderful afternoon.